Okay, so welcome back to this next video in which we are discussing Parkinson's disease and anti-Parkinson drugs. Okay, right, so we were just about to begin a discussion of the ubiquitin proteasome pathway, okay, within which the Parkin protein is going to be involved. Okay, I've said that Parkin is what's known as an E3 ubiquitin ligase enzyme. It's an example of an E3 ubiquitin ligase enzyme. Okay, so we want to see what that actually means, so we need to discuss this pathway, and I think we'll actually get a fresh sheet of paper for the discussion of this pathway. Right, okay, so the ubiquitin proteasome pathway then, it is a way of degrading proteins that either you no longer need anymore or that are broken in some way. Okay, so this is the basic idea, that firstly what you are going to do is you are going to tag the proteins with a certain molecular label, okay, which is going to be the label of destroy me. Okay, and this label is going to be recognized by a structure known as the proteasome, which is then going to do the job and destroy the protein. Okay, right, so we're now going to discuss the process by which this molecular tag is put onto proteins. Okay, right, so firstly, what is the molecular tag itself? Okay, so the molecular tag is ubiquitin. Okay, so ubiquitin is a protein itself, okay? It's actually a 76 amino acid protein. So 76 alpha alpha, what that means is amino acid. Okay, so let's me draw ubiquitin here. So let's say this is the amino group of ubiquitin. Okay, and we'll say this is the polypeptide of ubiquitin, so this represents the polymer of amino acids. It represents amino acid after amino acid after amino acid, and then you have the carboxylic acid tail of the ubiquitin protein. Okay, so I'll colour it in blue like so. So this is ubiquitin, and for short it's often abbreviated as UB. Okay, and there are 76 amino acids in this ubiquitin polypeptide. Okay, right, so let's say we also start off with some protein that we want to tag with the ubiquitin. Okay, so let's put it here. So we'll have this as the amino terminus of this protein, and this is the carboxylic acid terminus of this protein. And we'll colour in this protein that we are going to tag for destruction in red here. Okay, so this is going to be the condemned protein, the protein which is going to be destroyed. Okay, right, so how do we firstly attach the ubiquitin protein onto the side of this condemned protein? Okay, well, the first step in this process is going to involve firstly attaching the ubiquitin protein onto another enzyme. Okay, so onto a different protein. So the first enzyme you are going to attach the ubiquitin protein onto is what's known as the ubiquitin activating enzyme. Okay, so ubiquitin activating enzyme. Now in humans there is only one ubiquitin activating enzyme, so this is a single enzyme. Okay, and this has another name, it's also known as E1. Okay, and there is only one of these, so there is one E1. Okay, and this is going to be different for E2 and E3. E2 and E3 we're going to have loads of different types, but for E1 there is only one enzyme. Okay, so when I say E1, that means a specific protein within the human cell. Okay, so here it is, the ubiquitin activating enzyme. Okay, so I'll colour it in orange here. And the first step is going to be to attach the ubiquitin protein onto the E1 protein here. Okay, so in the active site of the E1 enzyme, you are going to have a phyl group here. Okay, so a phyl group is just a sulfur atom bound to a hydrogen atom. Okay, shown here. Now, if you are not familiar with the chemical properties of a phyl group, then there is a useful line that I can give you to give you more confidence with this chemical group. Okay, and the useful line is that these phyl groups have very, very similar chemical properties to alcohol groups. Okay, because sulfur is in the same group of the periodic table as oxygen. Okay, so sulfur has similar chemical properties to oxygen. Now, if that sulfuratom was an oxygen atom there, this would be an alcohol group. Now, I've just said that the sulfur atoms have similar chemical properties to oxygen, and therefore we would expect this thiol group here to have similar chemical properties to an alcohol group, and indeed it does. Now, why is this so important? Well, the way that you are going to attach the ubiquitin molecule onto the E1 ubiquitin activating enzyme is by binding this carboxylic acid group of the carboxylic acid terminus of the ubiquitin polypeptide onto this thiol group of the E1 ubiquitin activating enzyme. Okay, and the way that you're going to do this is by reacting this carboxylic acid group with the thiol group, and it's going to react in a very similar way to how a carboxylic acid group would react with an alcohol group. So if that was an alcohol group, then we would say that the carboxylic acid group would form an ester link with the alcohol group. Now, it's going to be pretty much that exact same reaction that's going to occur here, but it's going to be with a thiol group rather than an alcohol group. So we're going to form what's known as a thioester link rather than an ester link. Okay, so let me show this. So if I show my ubiquitin here, okay, here is the carboxylic acid group of my ubiquitin. Here is the rest of my ubiquitin. Here's the amino group. Okay, right, so I'll colour in ubiquitin here in blue. Okay, and what I'm going to do is I'm going to take the alcohol group off the carboxylic acid group. I'm going to take the hydrogen off the thiol group. I'm going to bind those two things together to make water, and then I'm going to bind the sulfur atom of the thiol group to the carbon of the carboxylic acid group, and this link is going to be a thioester link. Okay, and it's going to be a condensation reaction since it produces a water molecule here. Okay, right. So that's the first step in this ubiquitin proteasome pathway, that you firstly need to bind the ubiquitin protein onto an E1 ubiquitin activating enzyme. Okay, now, this reaction is actually dependent on the hydrolysis of ATP, because this water molecule here is going to be used to hydrolyze a molecule of ATP. Okay, so we're going to hydrolyze ATP to ADP and inorganic phosphate. So the first step is dependent upon the hydrolysis of ATP, or it's coupled to the hydrolysis of ATP, and this provides the energy for the binding of this ubiquitin protein onto the E1 ubiquitin activating enzyme. Okay, right. So the next step then in the ubiquitin proteasome pathway, and I might just label this up. This is step 1 in the ubiquitin proteasome pathway. Okay, so I'm just going to put the title because we lost that when we moved on to the other page. So this is the ubiquitin proteasome pathway, and this is an extremely important pathway as far as degrading proteins is concerned within cells. Okay, so the ubiquitin proteasome pathway. Okay, and I'll underline it in vivid purple because it's so important. Okay, right. So now let's move on to stage 2 of the ubiquitin proteasome pathway. Okay, so in stage 2, what's going to happen is we are now going to transfer this ubiquitin group off an E1 enzyme and onto an E2 enzyme. Okay, and this is a boring step in this process because all we are literally going to do is transfer it off one enzyme with a thiol group onto another enzyme with a thiol group. Okay, right, so let's show this. So this next enzyme is going to be what's known as an E2 enzyme, or you can also call it a ubiquitin conjugating enzyme. Okay, now, unlike E1, where there was only a single ubiquitin activating enzyme, there are multiple ubiquitin conjugating enzymes in humans. Not quite as many there's not quite as many E2 enzymes as there is going to be E3 enzymes, but there is more than one, certainly. Okay, so what is an E2 enzyme going to do? So basically, you're going to bring in your E2 enzyme here. So I'll show it here. Okay, and it too is going to have a thiol group in its active site. Okay, and the idea is what you're going to do is you're going to take the ubiquitin molecule from your E1 enzyme Okay, so I'll draw my E1 enzyme out again here. So this is my E1 enzyme, and I'll colour it in orange so that it's recognisable. Okay, and we now know that my E1 enzyme has this ubiquitin molecule bound onto it via this Thioester link, by which we've attached the carboxylic acid terminus of the ubiquitin protein onto the thiol group that's in the active site of our E1 enzyme. Okay, so here is our ubiquitin protein here. So I'll colour this in blue. Okay, and what we're now going to do is we're going to transfer this ubiquitin molecule onto this sulfur here, and then we'll put the hydrogen off this sulfur onto here to recreate the thiol group of the E1 ubiquitin activating enzyme, and then we've transferred the ubiquitin group onto the E2 ubiquitin conjugating enzyme. Okay, right, now I'll colour the E2 ubiquitin conjugating enzyme in red. And once again, I'll stress the point that whilst there was only one type of E1 ubiquitin activating enzyme, full stop, there are loads of different E2 ubiquitin conjugating enzymes. Okay, right, so that's step 2 in the ubiquitin proteasome pathway, that we need to transfer the ubiquitin molecule from an E1 enzyme onto an E2 enzyme. Step 3 then, and this is going to be a more interesting step. Okay, now what we are going to do is we're going to use the ubiquitin molecule that is on our E2 enzyme, and we're now going to transfer it onto our condemned protein. So let's bring our condemned protein in here. Okay, right, so firstly we need to discuss what are you actually going to attach the ubiquitin protein onto in the condemned protein. Okay, so we need to attach it onto the condemned protein, but how are we going to attach it onto the condemned protein? Well, you always use a certain type of amino acid residue. Now you might think, well, we just need to use an amino acid residue with a thiol group, and then there's only one with a thiol group, so it would be cysteine. But no, you don't use cysteine to attach ubiquitin molecules on. Instead, you use the amino acid lysine. Okay, single letter K. Okay, because leucine took L. Right, so let me show you the structure of a lysine residue, and then we'll discuss how we're going to attach the ubiquitin molecule onto the lysine residue. So, once again, I'll show it as a residue, so I'll show it as though it's bound within a polypeptide. So here's the amino group here. Here's the alpha carbon with a hydrogen coming off it. Here's the carboxylic acid group. And then the R group of a lysine residue consists of 4 methylene groups, and because I don't want to draw 4 methylene groups, what I'm going to do is I'm going to put one methylene group like this. I'm going to bracket it, and I'm going to subscript that 4, and that means repeat this 4 times. Okay, and then you're going to have an amino group here. Okay, so this is a lysine residue here. Right, okay. And now what we're going to do is attach on our ubiquitin molecule onto a lysine residue within our condemned protein. Right, now how are we going to do this? Well, what we're going to do is we're going to transfer the ubiquitin molecule off our E2 enzyme, and I'll draw the E2 enzyme with the ubiquitin actually attached on, because we haven't got that picture so far, we've just got this picture of it being transferred on. Okay, so we've got our sulfur atom here with the Thioester link onto our carboxylic acid group of the ubiquitin protein here. And here's the amino group. Okay, so the basic idea then is that what you're going to do is you're going to transfer the ubiquitin molecule onto the amino group of a lysine residue within your condemned protein. So you're going to bring this, and you're going to attach it onto the nitrogen. You're going to take a hydrogen off the nitrogen and attach that back on the onto the sulfur to recreate the thiol group on this E2 ubiquitin conjugating enzyme here. Okay, right, now let's put that in perspective on our condemned protein picture here. So the condemned protein will have some lysine residue here, which I'll denote as K. Okay, this lysine residue will have an amino group, so I'll actually show the amino group. And now we are going to attach by an amide link. Okay, so that's an important key word. When you attach a carboxylic acid group to an amino group like this, it's known as an amide link. Okay, so here's our amide link, and then we'll have our ubiquitin protein tagged on the end like so. Okay, right. So here we have now attached our ubiquitin protein onto our condemned protein. Okay, now an enzyme is required to catalyze this reaction. Okay, and the enzyme which catalyzes this reaction is then known as an E3 ubiquitin ligase enzyme. Okay, so this reaction is going to be catalyzed by a third enzyme. Okay, I'll put this here, which is called an E3 enzyme, or you can call it a ubiquitin ligase enzyme. And there are absolutely loads of different types of ubiquitin ligase enzymes. Okay, and what they differ in is which proteins they target, so which proteins they are capable of attaching ubiquitin molecules onto. Okay, so this is what Parkin is. It is a ubiquitin ligase. Okay, now before we actually say anything more about what Parkin is actually going to do, we need to complete this story, because even though we've now found what E3 ubiquitin ligases are, we haven't actually completed the ubiquitin proteasome pathway yet. Okay, so we have so far attached one ubiquitin molecule onto our condemned protein. Now, one ubiquitin protein molecule on the side of a protein is not enough to get this protein destroyed by the proteasome. Instead, you have to attach more than one ubiquitin molecule onto a protein to get it to actually be degraded. Now, you might think, well, that's easy, all we need to do is find more lysines and attach more ubiquitin molecules on, but in fact you don't need to find more lysines, because what you can do is use lysines that are in the ubiquitin molecule itself. Specifically, the ubiquitin molecule itself has a lysine at position 48, okay, so this is K48 here, okay, and what you can do is now attach another ubiquitin molecule onto the amino group of this lysine at position 48 on this first ubiquitin molecule. Okay, so all you need is another E2 enzyme which has got a ubiquitin molecule attached to it. And then you just need your E3 ubiquitin ligase to come along again and attach that ubiquitin molecule onto the amino group off the thiol group of the E2 enzyme. Okay, so here it is. So here's the carboxylic acid group attached via this amide link, and here's this next ubiquitin molecule here. And of course you can continue on. This next ubiquitin molecule, that will also have a lysine 48, and you can attach another ubiquitin molecule onto that, and so on and so on. Okay, and you can form what's known as a polyubiquitin tail. Okay, now in order to get a condemned protein to actually be degraded by the proteasome, it needs to have at least 4 ubiquitin molecules within the polyubiquitin tail. Okay, so to get a proteasome to take your protein seriously for degradation, it needs to have at least 4. Okay, but you know, you can have a huge number of these ubiquitin molecules tagged onto you in this polyubiquitin tail. It can go up to 50 or more, basically. So you can get massive, great ubiquitin tails, okay, attached onto proteins. So, now the final stage then, we'll put this as stage 4. Okay, so you've now attached on this polyubiquitin tail onto your protein, and now what's going to happen? Well, proteins that get these ubiquitin molecules attached onto them in this way are destined for the proteasome. So what is the proteasome? Well, basically the proteasome is quite a formidable thing in molecular biology. Okay, it is a tube. Okay, and proteins go in one side, and fragments of proteins come out the other side. Okay, so basically, proteins that get large polyubiquitin tails attached to them end up attaching to the mouth of the proteasome. Okay, so the polyubiquitin tail targets the protein to the proteasome. So if we have our condemned protein here, let's then have our ubiquitin tail here, and I'll just put 4 of them on just because that's all I can be bothered to draw. So here are my 4 ubiquitin molecules attached on in my polyubiquitin tail. Okay, they are going to bind to the mouth of the proteasome, okay, the entrance of the proteasome, and then what's going to end up happening is that condemned protein here in red is going to end up being unfolded, and it's going to end up being pulled through the tube, okay, and the tube contains a huge number of protease enzymes, okay, that line the walls. And these protease enzymes are going to break the protein apart. They don't quite break it down into amino acids. They break it down into little fragments, okay, so a few amino acids joined together. Okay, and that's how you destroy proteins, well, one of the mechanisms by which you destroy proteins. Okay, right. So, Parkin then. What does Parkin do? Well, Parkin is an E3 ubiquitin ligase that does this for alpha-synuclein. Okay, so it is responsible for putting ubiquitin groups onto alpha-synuclein, and therefore it's responsible for targeting alpha-synuclein for degradation. We have said that Parkinson's disease it consists of this neurodegeneration. Okay, it's because of this neurodegeneration that results from alpha-synuclein accumulation. Okay. If you can't destroy the alpha-synuclein, then it makes sense that alpha-synuclein will aggregate. So that is potentially how Parkin mutations are causing this very severe form of Parkinson's disease that is hereditary. Okay, if you've got no Parkin, you can't destroy the alpha-synuclein anymore because it can't be destroyed by the ubiquitin proteasome pathway, and therefore it's going to accumulate within your cells.